Welcome to the SDK Health Sciences Biochemistry classes. Today our topic is hemoglobin. Hemoglobin is an oxygen-carrying pigment of the red blood cells. It is a globular protein that is comprised of heme and globin. Globin is a simple protein, whereas heme is a combination of ferrous form of the iron and porphyrin. So in a nutshell, we can say that hemoglobin is a combination of heme and globin. Globin is simple protein. Heme is a combination of iron and porphyrin. The oxygen binding capacity of the hemoglobin is 1.34 milliliter of oxygen per gram of the hemoglobin. Some more few important points about the hemoglobin. Hemoglobin is present in red blood cells. The average normal red blood cells count is about 5.4 million per microliter in men and 4.8 million per microliter in women. Each human red blood cell contains about 29 picogram of the hemoglobin. So in total, there are about 3:10 raised to the power 13 red blood cells and about 900 gram of the hemoglobin in the circulating blood of an adult man. The normal total circulating volume is about 8% of the body weight, which is about 5,600 milliliter in a 70 kg man. The normal value of the hemoglobin in male is about 16 gram per deciliter, whereas in female it is 14 gram per deciliter. Structure of hemoglobin. Hemoglobin is comprised of two components. One is heme, another is globin. Heme is an iron porphyrin compound which is comprised of porphyrin and ferrous form of the iron. Porphyrin is comprised of 4 pyrrole rings, and these 4 pyrrole rings, they are linked together with methynyl bridges, with methynyl bonds. This is CH double bond. However, the globin is a simple protein and is comprised of, in hemoglobin, is comprised of 4 polypeptide chain, 2 alpha and 2 beta. Now the component of the porphyrin ring. Porphyrin ring is comprised of 4 pyrrole ring. And I can make pyrrole ring here. This is the pyrrole ring. Let me make it a little bit more clear for you guys. Like this. And then here is nitrogen, like this. So this is the pyrrole ring. It has 2 bonds, 2 ends. One end is that end which is towards the nitrogen, and another is the outer end. So I classify this end like this. That one which is the nitrogen side, another is on the other side. So these are the pyrrole rings. Let me make it more clear. First make nitrogen, then like this. My drawing is not good, but anyway, I will try my level best to make it easy for you. Like this. So these are the 4 rings. Now before going to the detail of these 4 rings, let me give you few important points about these 4 rings, or this pyrrole ring. Number 1, as I said earlier, I describe it in this way, that it has an inner ring and an outer ring, or an inner bond or outer bond. This bond, I call it an inner bond, where it is attached to nitrogen. And this bond which is on the outer side, I call it the outer end or outer corner. Now with the help of these inner bonds, these pyrrole ring, 4 pyrrole ring, they are designated as from A to D. They are connected with each other with the methine bridges. However, the other outer ends have attached to some other important groups. So inner, and because they are four in number, so the inner are 8, and we count them from as like 1 prime and 2 prime, whereas outer are number as 1 and 2 directly. So you need to keep this in your mind. Now some other calculations. We have 3 other groups attached to these 4 rings. Number 1 is a methyl group. Number 2 is a vinyl group. And number 3 is propionic acid group. Methyl group is CH3. It is attached to position 1, position 3, position 5 and position 8. So 1, 3, 5, 8. Keep in mind that 1, 3, 5, 8 are the outer ends, outer bonds. Vinyl group CH, double bond CH2 attached to 2 and 4th position. Propionic acid CH2, C double OH and is attached to 6th and 7th position. Keep in mind that these are the outer bonds. And then in these 4 structure, these 4 bonds, they have alternative double bonds. So let's see in detail of these. So these are the 4 rings, 4 pyrrole ring A, B, C and D. Now these are the inner rings: 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, 6 prime, 7 prime and 8 prime. With the help of these inner bonds, with the help of these inner corners, these 4 rings, they are attached to each other. With the help of these 4, they are attached to each other. With the help of these 4, they are attached to each other. Like this. So you can see that this is a ring now. The ring is complete. And with the help of these 4, they are attached to each other. These are the methane bridges. Now let's put a double bond in it, or we can give number to the outer ring now. This is 1, this is 2, this is 3, this is 4, this is 5, this is 6, this is 7, this is 8. 8 bonds are there. 4 rings, 8 bonds are there. Now let's put double bond in it. Alternative double bond in it. So once we put alternative double bond, then the methane bridges will be complete. So this is a double bond. There is no bond here. Then there will be a double bond here. There will be no bond here. Then there will be a double bond here. There will be no bond here. Then there will be a double bond here. There will be no bond here. No double bond. Bond is there but no double bond. There will be a double bond here. Then no double bond here. Then there will be a double bond here. And there is no bond here. Then there will be a double bond here. And there is no double bond here. Then there will be double bond here. And then there will be no double bond here. And then last but not the least, there is a double bond here. Clear? And even then here, there is no double bond here. And this is the last, this is the last one. Last but not the least, this is the no double bond here. So you can see that these are the alternate bonds. One double bond, no double bond here, then there is a double bond here, then there is no double bond here, then there is double bond here, then there is no double bond here, then there is double bond here, then there is no double bond here, then there is double bond here, then no double bond here, then there is double bond here, then there is no double bond at this position, then there is double bond here, then there is no double bond at this position, then double bond here, no double bond here, then double bond here, no double bond here, and last but not least, this is the double bond here. Clear, guys? I hope, I tried my level best to make it simple for you. Now here you can see these are the bridges are complete. The methine bridges are complete, and this is a complete ring. This is a complete ring. If let's see, we can fix it here, then you can see here there will be iron in the form of ferrous here, Fe plus 2, and it will attach to 4 nitrogen. It will attach to 4 nitrogen with its 4 valencies. It has 6 valencies. Iron has 6 valency, and with the 4 it is attached to nitrogen of the pyrrole ring. Now let's come on and add the other things in the structure, the other few points that we have seen in the previous session. So if you want, I can give you a quick feedback of this. So we have 4 pyrrole ring, A, B, C, and D, and we divide the pyrrole ring into 2 components. One is the inner component, another one is outer one. And in inner we count them 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, 6 prime, 7 prime, and 8 prime. And the outer one, they are called 1, 2, 3, 1, 2, 3, then 4, then 5, then 6, then 7, then 8. The pyrrole rings, they are attached to it with each other with the help of the methine bridges, the CH bridges, and then we put double bond, alternative double bond, and the bridges are complete. The bridges are complete. Now let's see the other thing. So let me give you a quick review of this. So these are the bridges here. This is the bridge here. This is the bridge here. Here is the bridge here, and here is the bridge here. Then we have alternate double bond, no bond here. There is a double bond, no bond, double bond. This is a double bond. There is no bond, double. This is double bond. There is no bond. There is double bond. This one no. There is double bond. This is not. This is double bond. This is not. This is double bond. This is not. This is double bond. This is not, and this is double bond. And in the outer ring, we will do some calculation here. 1, 2, 3, 4, then 5, then 6, then 7, and then 8. Let's add the groups, methyl groups, and we know that methyl group would be added at 4 position. Position 1. So here CH3. Then position 3. See here, this is the position 3. So this is CH3. Then position 5. See, this is position 5. 1, 3, 5, 8. CH3. And then position 8. This is position 8. CH3. So we added the methyl group. Then we have 2 vinyl group, that is CH double bond CH2. And these vinyl groups, they are attached to position 2 and position 4. So here CH double bond CH2. Then position 4, here CH double bond CH2. Then we have 2 propionic acid, CH2, C double bond, COOH, and it is attached to position 6. So this is CH2, then CH2, then COOH, and then position 7, CH2, then CH2, then COOH, COOH. And then last but not the least, iron, as we put it earlier here. So the pyrrole ring is complete. The pyrrole ring is complete. I mean, it's very simple and doable. A, B, C, and D. This is the review. You can see here A, B, C, and D. These are the methane bridges. Methane bridges. This is CH3 at position 3, at position 1, at position 3, at position 5, and position 8, 1, 3, 5, 8. CH3, 1, 3, 5, 8. Methyl group. Vinyl group, CH, C double bond H2, position 2, position 4. Propionic acid, position 6, position 7. And alternate double bonds. Globin. Globin is a protein, simple protein, and the protein contains large quantities of two important amino acids. One is histidine and another is lysine. It is produced by usual mechanism of the protein synthesis. Usual mechanism of the protein synthesis is DNA to RNA, and then from RNA to protein synthesis. This job is called transcription, and this portion is called translation. The globin chain here in hemoglobin is comprised of 4 polypeptide chain, 2 identical alpha chain, each is 141 amino acid, and 2 identical beta chain, each is 146 amino acid. Each polypeptide chain contains a heme molecule. So we have 4 pyrrole rings and 1 heme molecule. One heme molecule. So one hemoglobin molecule contains 4 heme units, and it is shown that heme stimulates the globin synthesis. So for example, this is one chain, this is another chain, this is another chain, this is another chain. They are interconnected with each other, and they have salt bridges in between. They are interconnected each other. This is alpha 1, this is alpha 2, this is beta 1, this is beta 2. And then in them they have heme pocket. The alpha subunit heme pockets, they are open. However, the beta subunit heme pockets, they are closed by an important amino acid, valine. So this is the iron, which is attached to 4 nitrogen groups. Iron is attached to 4 nitrogen groups, as we saw in the previous thing, and it is attached to the 5th valency of the iron, attached to histidine, component of the globin of the polypeptide chain of alpha and beta. Then the pyrrole ring, 3rd pyrrole ring and the 4th pyrrole ring, the position 6th and position 7th, where they have propionic acid. These propionic acid, they are also connected with the polypeptide chain, alpha beta polypeptide chain. So the connection of heme iron component of the ferrous form of the heme connected with the globin molecule by 3 connections. 1 with the iron valency directly, and the 2 with the propionic acid, So 1 is the iron directly, and 2 with the propionic acid, The 6th valency of the iron, with the help of the coordinated bond, it is connected with the oxygen molecule. It is connected with the oxygen molecule. If you see the functional form of the hemoglobin, there are two functional forms of the hemoglobin. One is taut form, another is R form. The taut form is called deoxy form of the hemoglobin, and it is called taut or tense form. The taut form has low oxygen affinity, and the R form is called the oxy form of the hemoglobin. It is called R or relaxed form, in which the polypeptide chain have more freedom of the movement, and the R form is the high oxygen affinity form of the hemoglobin. Let's see what do you mean by taut form and the R form. So the taut form, these are 4 chain, 2 alpha 2 beta 2 alpha 1 alpha 2 beta 1 beta 2 chain. And as I said earlier, the heme pocket in the alpha subunits, they are open, whereas the heme pocket in the beta subunits, they are closed by valine amino acid, by amino acid valine. They are interconnected with each other, and they have methyl salt bridges in between them, many salt bridges in between them. So when they have many salt bridges in between them and there is no oxygen here, the heme pockets are empty, and affinity for oxygen is low. So this structure is known as taut form of the hemoglobin. More salt bridges, heme pockets empty, and the affinity for oxygen is low. So this structure is known as taut form, Ta, or taut form, tense form of the hemoglobin. As soon as oxygen arrives and it is taken up by the heme pocket, which is present in the alpha subunit, because as we said that alpha subunit is open unit, it is accepted by the alpha subunit. So by accepting oxygen, the alpha subunit brings some conformational changes in such a way that it performs two functions. Number one, it breaks the bridges, the salt bridges in between. So the structure become relaxed. That's why it's called R form of the hemoglobin. And second, the affinity for oxygen increases. And then the movement, the structural change in this molecule will turn open the heme pocket in the beta subunit as well by removing the valine. The valine is removed. So as such, the valine is removed, then the heme pocket become open, and then oxygen comes and enter here. So in this case, the R form has 4 molecules of oxygen. The first two come to alpha subunit. The alpha subunit brings some conformational changes. These conformational changes remove the valine of the heme pocket in the beta subunit and allow oxygen to enter into the heme pocket of the beta subunit. This conformational changes also break the bridges, the salt bridges between the two units, and then the structure become relaxed. I hope it will be clear up till now. If there is any question, any comment, please give me your feedback. Your feedback will help us to improve our sessions. Thank you very much and best of luck.